Professor at Division of Medical Oncology. He is a recipient of many awards. Dr. Singh has also filed 2 patents on Mycobacterium tuberculosis. He is a fellow member of many prestigious academic societies. Dr. Singh has published more than 60 quality research articles in peer-reviewed journals of international repute. He has also edited 5 books, authored 4 books, and also contributed authors more than 30 book chapters published under Springer, Taylor and Francis elsewhere. He has also trained many postdoctoral, doctoral, postgraduate and graduate students and currently supervising 7. He has also edited 5 books, authored 4 books, and also contributed authors more than 30 book chapters published under Springer, Taylor and Francis elsewhere. He has also trained many postdoctoral, doctoral, postgraduate and graduate students and currently supervising 7 doctoral, 2 postdoctoral and 4 MTech students. Published under Springer, Taylor and Francis elsewhere. He has also trained many postdoctoral, doctoral, postgraduate and He has also trained many postdoctoral, doctoral, postgraduate and graduate students and currently supervises 7 doctoral, 2 postdoctoral and 4 MTech students. Thank you, Professor Saktivel and Professor Indrakant for being with us today. Over to you, sir.
Thank you, Dr. Silpi, for such elaborative introduction for chairperson as well. I think we must elaborate introduction to our esteemed speakers, invited guest for this session. We have 3 speakers in this session: Dr. Neha Kaushik, Professor Sujit Kumar Ghosh, and Professor Nagendra Kumar Kaushik. So let's start with Dr. Neha Kaushik. Before Madam is going to start her invited talk, I would like to introduce to this our audience. Dr. Neha Kaushik is an accomplished professional currently holding the position of assistant professor in the Department of Biotechnology and Marketing at University of Suwon, South Korea since 2020. Prior to this, she served as a research professor and successfully led scientifically independent research projects from 2015 to 19 at Department of Life Science, Hanyang University, Seoul. Her research interests are primarily focused on cutting-edge areas such as biomaterials, oncogenes, nanomedicine, plasma medicine, cancer biology, immune modulation. Dr. Kaushik's scholarly facts is remarkable with over 87 SCI papers published in prestigious journals. Dr. Kaushik is actively involved in scientific publications and editorial activities. She served as an editor for esteemed high-impact journals. Her impressive publication record, editorial roles in reputed journals, and leadership in independent research projects underline her standing as distinguished figure in the scientific community. Over to Dr. Neha Kaushik. Thanks.
Thank you so much, Professor, for the nice introduction. So, am I audible to all of you? Am I audible? Hello?
Thanks.
Yeah, am I audible to everyone? Professor, for the nice introduction. So, hello?
Audible. Yes, you are audible.
Okay, thank you. Yeah. First of all, I would like to thank the organizing committee for providing me such a great platform, and it was nice to see you all. So, could I start my talk now?
Yeah. Yes, you are audible.
Okay, thank you. Yeah. First, I would like to share my screen. I cannot share my screen. So could you please allow? Hello? I cannot share my screen. It is first identified is mid-1960s. So basically we have looked for what is the research question here in our work. Next, please. Yeah, so research question is actually the 2DG is very well known for the as an anti-cancer drug before, and people already know that 2DG can block the antiviral infections also, but eventually people doesn't know what is its impact on the host defense response. It means host immune response. It can elevate or it can improve. The people doesn't know about it, so we are working to look for both aspects. It can block the viral infection as well as can improve the host defense response itself. So here our question is, can 2DG enhance the host defense response while simultaneously inhibiting the viral replication for the antiviral response? So we basically kind of dual response we are looking for in this work. Next one. Yeah. So firstly, we have confirmed that ICHE-2023 virus can selectively increase the glucose consumption and ATP generation in the host cells. So for before using the 2DG, we want to confirm that what is the response of this virus towards the glucose consumption and the ATP in the host cells. So we have infected the normal lung cells and the epithelial alveolar epithelial cells, which is a respiratory, which is very important for the respiratory system. So we have checked the both of the cell type. We have seen the increased glucose uptake, as you can see by the FACS analysis, and also the confocal microscopy confirms the elevated glucose level in the infected cells itself. And to look further and further confirmation, we have checked the glycolysis markers, which is the GLUTs receptors, which is very important whenever we're looking for the glucose uptake. So GLUT levels was quite enhanced in the infected cells, and also the ATP was quite increased. So this is we confirm with the both cell types, MRC5, which is the normal lung cells, Kalus cells, this is the alveolar epithelial respiratory cells. And also the similar profile we have observed in the both cell types in case of the ATP also. So we conclude that upon the virus infection, the both of the cell type can increase the glucose consumption and the ATP generation. Yeah, next slide, please. Next slide. Yeah. Next, we want to look for the effect of 2DG against the 229E virus. So we have checked the EC50 and CC50. CC50 is a cytotoxic concentration where the 50% cells are viable. And about the CP, which is the, CP is the EC50 where effective concentration of 2DG against the viral infection. So based on this, we can calculate the selectivity index. Selectivity index is a therapeutic index, which measures the window up, through the EC50 and CC50. And based on this, we can check that if it is this drug can be pharmaceutically is acceptable or not. So here we have calculated the SI index, which is calculated as a 10.4 in case of the 2DG. This result we have seen in the MRC5. Yeah. And in this slide, you can see the CC50 of this virus is about 47.7 and EC50 results like a 4.75. So about the 4.75 millimole, the concentration is quite effective against the viral infection, and 47.7 millimole concentration is quite effective. I mean, it is not toxic towards the normal cells. So we can calculate the SI index based on these two values. Yeah, one more thing, this measurement was done after the 48 hours of infection. Next slide, we have here we have checked particular that how 2DG behaves towards the 229E replication process and its infectivity potential. So I think everyone knows the virus, every virus mostly contain the S spike protein, M proteins, and E proteins. So these individual proteins are responsible for the virus assembly. So firstly, we want to check the replication process. So for this one, we have checked the RNA expression level of the M, E, and N gene. And interestingly, we have observed that 2DG treatment can suppress the suppress the level of these genes in the infected cells, which is quite promising. And based on this, we can say the replication process of this virus after the 2DG treatment is quite slow, and 2DG can block the viral replication process. And on this slide shows the S protein expression level by the confocal microscopy and the FACS analysis, because the S protein is quite important whenever they have to do infection, because it's a surface protein. So S protein level was also suppressed, as you can see in the pictures quite well, that upon the 2DG treatments, as a quantitative we have observed by the FACS analysis, that S protein level is quite decreased after the 2DG treatments. It means the 2DG treatment can suppress the infectivity of this virus inside the host cell. Next slide, please. Yeah. So next we have checked the 2DG treatment can improve this cytopathic effect, which is confirmed by the CP. So we have checked with the different concentration of 2DG also. So we have observed that upon the 10 millimole concentration, about the 2.57 log order difference we can observe in the viral infection level. So this is a kind of one measurement which is widely used to check the viral infection process. So this is we call TCID50. So TCID50 results, if you see the table, you can see the control has a 10 log or 5.6, and here is a 6.6, and the 10 millimole have a 4.03 log order difference. But when you compare those two, and you can calculate, there is a difference of 2.57 log order difference between those two samples. So we have checked different concentration also, but this is quite good effect we have observed. If you see the graphs, then after the 10 millimole, there was a quite good difference we can observe, that 2DG at 10 millimole concentration is also quite well. And this concentration is also not toxic to the normal cells, so we can safely use this concentration of 2DG if you want to look for the further experiments. Yeah, next, please. Yeah. One more slide. Yeah, next. Yeah, here. So, no, please go back. Yeah, here. So in this slide, basically we want to check that what is the effect on the cellular death and stress response, because whenever we have the virus infection, the cells get stressed, and because of the stress, cells have the death. So we have checked the particular cell death phenomena. If you see the FACS analysis, you can see the PI proportion of the PI cells got reduced after the 2DG treatments, and the live or dead cell staining also confirms that dead cell population was quite decreased after the 2DG treatments. It means whenever we give the 2DG to the infected cell, the cell can relieve from the virus-induced stress, and eventually the cell death goes down. So this data confirms this phenomena, and based on our hypothesis, we can confirm that 2DG can suppress 2DG can suppress the cellular death itself after the infection in the normal cells. Yeah, next. Next, please. Yeah. So finally, what is our final goal about the host immune response? So we have checked the host defense response related genes. Go back to the earlier slide, please. Yeah, here we have checked the different immune related genes, which is the interferons also, and G84, AB69. So these are quite different signaling which are related to the immune defense responses. And after the 2DG treatment, their level was quite improved, but some of them are quite highly induced. So we can say that some of the markers are quite well increased after the 2DG treatment. So we can assume that 2DG treatment can improve the host defense response itself, even after, together with the replication process inhibition. So when our final goal was to check the glucose level, next please, glucose level, next please, yeah. Our final goal was to check the GLUT levels also, because in the first slide we have shown that after the infection, glucose level was increased, so GLUT also increased and ATP also increased. So we have to check the after 2DG, this effect was reversed. As we can see with the mRNA expression, the GLUT was suppressed and ATP also suppressed. So this means we can eventually block the viral replication process, and also at the same time we can improve the host antiviral responses. Yeah. So the summary of this talk is, so we suggest in this work that very low doses, that is the 10 millimole concentration, can inhibit the glucose uptake, which indicates that 2DG consumption is the viral infected host cell was mediated by the high affinity glucose transporters whose level was amplified upon the coronavirus infection. So the main part of this study, we offer the new understanding of the molecular action of the 2DG to improve the host defense response in the coronavirus infected cells, which has not explored so far. So this work we have published recently. Yeah, next slide. So yeah, this is one of the same extended work we have performed, where we have used the gas plasma, which maybe you can see, you know or not, I don't know, but this is the first state of matter. This is where we have the gas plasma. It is the medical plasma. So here we have, when the plasma generated, there are some reactive species formed. So basically in this work, we have used the metabolic altered cells, and on the other hand, we have used the viruses which are treated with the plasma. So we have checked this combination, and we have observed the plasma treated virus can improve the CPRR, this is cytosolic pattern receptors, and the induction of the CPRR can induce the ISG in the host cells, and they can eventually block the viral infection in the metabolic altered cells. So this is one of the work which is the extended part of this work. And this slide has a collaboration which I have the overseas and the domestic so far. So this is some of the universities which I work locally and overseas. So thank you very much for providing me this opportunity to present here. Some of the part I skipped because of the time limit. Thank you very much. If you have any question or comment, please feel free to ask.
And now the session may be open for one question. Any question from audience? Dr. Neha, I have.
Yeah, sure. Hello, you're not audible.
Can you hear?
Yeah, I can hear now. Yeah.
Yeah. Normally, those who suffered by COVID, their immunities goes down. So what is the major issue with that? Even after they recover, after a few years, their immunity is very poor. Why it is?
That is the post-COVID issues, right?
Yes. Whether this 2G treatment, whether it will help on that?
Actually, I'm not sure on this part, because but just in this work we have looked for, because 2DG is already people are using for the antiviral as an antiviral drug in India. So, but nobody has a claim that it can improve the immune system itself at the same time. So we believe that afterwards, we don't know, but of course, that's a good question. We have to look for that. And
Okay.
Unfortunately, I cannot answer at this moment this question.
Fine. Anyway, thank you, Dr. Neha, for the wonderful lectures. On behalf of organizing committee, we thank. Yes, we thanks for your valuable times. Let us thank Dr. Neha with
Thank you, and sorry once again for this slide things. Sorry, yeah. Thank you
Let us
once
move-
again.
Yeah, thank you. So let us move on to the next speakers. Our next speaker is Professor Sujit Kumar Ghose. He is a professor from Indian Institute of Science, Education and Research, that is ICHE, Pune. He did his postdoc from Kyoto University, Japan in 2009 and PhD from IIT Kanpur. He has achieved a distinguished award from various academic organizations such as Chemical Society of Japan Lecture Award, India Research Excellence Citation Award, Aura Young Scientist Award, and many more. He is a lifetime member of Chemical Research Society of India, Material Research Society of India, Society of Material Chemistry. Professor Sujit has supervised more than 20 PhD students, and his area of research is functional advanced porous materials. Example like metal organic porous, metal organic framework, porous organic polymer, porous hybrid composite material, on hazardous pollutant capture for safe drinking water. So he has more than 150 publications in reputed journals, many book chapters and review articles. With this brief introduction, I welcome Professor Sujit Kumar Ghose, and now over to you, Professor Sujit. Professor Sujit has some issue. He has joined and he left. So maybe we'll wait for him. Meantime, we'll go for our next speaker, Professor Nagendra Kumar Kaushik. Professor Nagendra Kumar Kaushik, are you ready?
Yes, hello everyone.
Yeah. So we have Professor Nagendra Kumar.
Your sound is not audible.
Automatic mic is off, getting off.
Yeah, now it is audible.
Okay, fine.
Professor, now it is audible.
Fine. There is some issue with the mic. Okay, no issue.
Yes.
So, Professor Nagendra Kaushik is from Wongon University, Seoul, South Korea, and holding the position in the Department of Electrical and Biological Physics and Plasma. And his profound focus on plasma bioscience, plasma agriculture, environmental, nanobiotechnology, and biomaterials. His groundbreaking research has resulted in over 200 publications in esteemed journal. Professor Nagendra has applied numerous patents and served as an editor for more than 25 esteemed journals. His exceptional contributions have earned with him well-deserved place in Stanford University and elsewhere. Prestigious list of world top 2 scientists continuously since 2021. With this brief introductions, I welcome Professor Nagendra Kumar Kaushik for the forum, and over to you, Professor.
Thank you, sir, for a very nice introduction. Can you see my slide?
Yeah, you are audible.
Okay.
You are audible. You may proceed.
Yes. Yeah. Hello, everyone. Myself, Nagendra. Actually, I am alumni of Delhi University. So Parthiba and all organizers are my friend eventually. So they invited. I really want to thank first of all these team members who organized this conference. And myself is working in South Korea since from last 13 years. I'm working in Kangwon University in Seoul at Department of Electrical and Biological Physics. and Plasma Bioscience Research Center. And I am also leading this center with the many departmental as well as university as well as NRF fundings. And we are working on several topics. First of all, I want to tell you about the non-thermal gas plasma. So we are using non-thermal gas plasma, and we are using it for biosciences, medicine, agriculture, or environmental issues. So today I will give you a little bit glance of this all topics in specific time. So I will talk about pathogen inactivation, like how we can inactivate pathogens such as viruses by using plasma. And I will tell you about some issues related to bacteria. So the biggest issue is dormant bacteria. So I will give you the glance of dormant bacteria, how we can treat them, and what are the issues with those bacteria. And second topic is related to the green and smart agriculture. And another topic is related to the plasma-based nanoparticle green synthesis. So this is basically Plasma Bioscience Research Center. This center is established, basically it's a long story. The basic laboratory is started in 1989 by a professor who is a leading scientist now in Kangon University. And after that, we opened many centers, and this Plasma Bioscience Research Center is opened in 2011 with the help of Government of Korea. And we have more than 110 members, and we are working on several topics related to treatment of cells, cancers, or treatment of proliferation of stem cells or proliferation of immune cell, etc., etc. Also, we are working on many biochemistry topics or chemistry topics, such as we are using various kind of reactive oxygen and nitrogen species for the synthesis of nanoparticles, as well as virus inactivation, for modification of blood, as well as modification of organic molecules. And eventually our goal is biology and environment, how we can use this plasma for some biological purposes, as well as how we can eradicate environmental issues by using these kind of plasmas. So this is the very basic definition of plasma. Simply says, commonly we can say plasma is 4 state of matter, like solid, liquid, gas. Solid is the first state of matter, then liquid, then gas. And if you give little bit energy to this gas, you can make plasma. So simply, this is the very simple definition. Other details, deep details, I will not explain in this presentation. So if you ask about the definition of plasma to me, I will just say the plasma is the 4th state of matter, and it consists of ions, electrons, reactive oxygen and nitrogen species. These are two words, ROS and RNS. So reactive oxygen nitrogen species we have in plasma, and little bit amount of UV that cannot harm to the people, and very little heat in some cases, not all cases. So these are the type of devices we have of plasma. You can see the different kind of devices, like physics people can understand it. It's very hard for the chemistry people to understand all these configurations. So we have different kind of devices such as floating DBD, facing DBD, coplanar DBD, floating jet, plasma jet, direct case, and plasma jet indirect case. So these are the devices commonly available in all over the world. But for your knowledge, I am telling you the whole universe, whole of our universe is 70% is made up of plasma outside Earth. And the common example of plasma in nearby you or neighboring to you, you can see the welding machine. Welding machine is basically hot plasma. So we are making cold plasma by using electricity as an energy. And plasma you can make at your home also. You just need to make 2 electrodes, high voltage electrode and the ground electrode. And in between 2 electrode, you can make plasma. This is only the principle to make plasma. There is nothing like magic. So plasma you can make at home also. You can make your own devices. You can tune your device. You can configure more to make it more advanced. That's depend on you. And by plasma, what you can do? This was maybe the question in your mind. So plasma on the surface can have electrons, and can make by these electrons, by this electrical energy, you can also have atomic species such as nitrogen, atomic nitrogen, atomic oxygen. And further, these atomic species and electron can react with the atmospheric air, such as water, oxygen, and nitrogen, to make many kind of reactive species, such as nitric oxide. That is very, very important. NO2, NO3, and other are like H2O2, ozone, singlet oxygen, and many others. So you can control this reactive species also as you want for your purpose. So nowadays we are focused on NO, so we are just making NO by tuning our plasma, nitric oxide. And further, these secondary reactive species react with the water, or your target can make many kind of other species such as N2O5, HNO2, HNO3, peroxynitrile, and etc., including S2O, S2O2. So these species can be used for some specific purpose. If you want to kill bacteria, if you want to inactivate viruses, you can just use harsh species. But if you want to protect human cells during this treatment, you can use NO. This is the one kind of strategy we can have. And we can control these reactive species for specific purpose, for specific synthesis, or for specific biological treatments, or specific environmental eradication of environmental issues. So these are application of plasma. These are some of application, not all. We can use plasma for medicine. First old application is sterilization of medical device. It is more than 300-year old. So before 300 year, people used to use this plasma device for sterilization of medical devices before surgery. And nowadays also in hospital, we have plasma sterilizers. And second application is you can treat cells, you can treat skins, you can treat organs, tissues, etc., stem cells, immune cells. And environmental application, obviously you can clean water by plasma. Already in market, there are some filters nowadays with the plasma technology, and you can clean exhaust gases. So if you talk about environment, you just think about soil, water, and air. So you can clean all these three things, soil, water, air, by using plasma. And in case of agriculture, this is very subjective. You can clean your vegetables, you can clean your fruits by the plasma devices. You can put plasma device at your kitchen to clean your vegetable and fruits, to disinfect them. And as well as it can be used in the field by the farmer for nitrogen fixation, for many other things. So that is very, very subjective area. And in dentistry, yes, we can clean devices like medicine, and we can treat teeth directly. We can treat gum, we can treat nerves, which are which have many big problems related to nerve or pulp or gums in dentistry. So today I will tell you a little bit about the coronavirus, how we are doing here, and how we are treating, like inhibiting coronavirus by plasma. So this is the recent report before 3 years in Trends in Biotechnology by our collaborators. The type of viruses we have, you know that we have DNA viruses, RNA viruses, now single-stranded RNA, double-stranded RNA, or DNA, like enveloped, non-enveloped, animal or plant viruses. So all those viruses can be treated by the plasma. Second thing, how the ROS and RNS generated by plasma have main mechanism. These ROS and RNS are responsible for inactivating these viruses. Basically, they are targeting, these RNS targeting viral nucleic acid and proteins to damage them, to make them ineffective particles. And by these ineffective particles, any human cannot be infected, or any plant cannot be infected. This is very, very simple mechanism. And in PBRC, in our center, we are focused on the 3 strategies. One, virulence factor, how we can control virulence factor of viruses by plasma. So in case of SARS-CoV-2, as spike protein, if you know that spike protein was the virulence factor. Similarly, in other viruses, we can have various kind of other virulence factor. We can target those virulence factor by using plasmas. We can directly damage them by the biocompatible RONS, these virulence factor. And second is virus entry. Can we block virus entry or binding of virus to the human cells? That is another topic. And for those cases, we are working on some kind of receptors available on human cells, how we can modulate them by using plasma. And third topic and most important topic is host cell response. Virus infection is okay, but what is going on behind this all scene of infection? It's about like immunomodulation, it's about the lung rejuvenation, it's about the pulmonary vasodilation. All these important topics are involved in the COVID virus treatment strategies by many hospitals as well as clinicians. So finally, we can control viral pathogenicity by using all these three strategies by using plasma. Recently, we did one thing. We made a plasma machine, which is based on the microwave. This microwave machine is just like you can think microwave you use at home, 2.45 gigahertz microwave. So we made a plasma with the help of microwave to make a pure, very stable nitric oxide gas to make nitric oxide water. So this strategy is also very fine. We can use this bottled water, nitric oxide bottled water, for the treatment of viruses. So we got we tuned it for very high concentration and we got it recently. This paper is already published in Bioactive Material. Its impact factor is 19. And recently, before 7 years, we published this paper. This NO water we can use to inactivate viruses. As Professor Neha in previous presentation said, we can measure two things if we want to check the virus is killed, like inhibited or not by our drug or strategy. So we can check the CP, cytopathic effect, how much virus is killing human cell. This is cytopathic effect. So we can check that percentage and we can say that, okay, our treatment is in reducing the cytopathic effect. It means the virus is live or dead. And another thing, we can measure the toxicity on the normal human lung cells in case of coronavirus. So whether our drug is killing the normal cells or not, that is another point. By these two things, we can measure SI index, that is selectivity index, and it is a therapeutic value of any treatment object or drug, or it can be radiation, it can be physical or chemical agent. So if SI index is high, it means your drug or your treatment object is very nice. If SI index is less, it means you cannot use it in market. So in case of, if you see here, this table, we use NO water, we use by plasma, and we use chloroquine, that is the positive control drug already available in market, hydroxychloroquine. That is also, that drug was also famous during the COVID time. And another drug is ritonavir, antiviral drug. And we found that our NO water is more better than the chloroquine and ritonavir. That is because our NO water is not toxic to the human cell. But this chloroquine and ritonavir are very toxic to the human cells at low doses of their low doses. So this is the therapeutic value, therapeutic validation of NO water. And we also, as a chemistry point of view, we checked it. We characterize our NO water by gas FTIR, not solid FTIR, not liquid FTIR. We have gas FTIR to check the species. And in this case, we found that the main species in our plasma was NO, not CO2, not any other gas or molecule. And this is the graph. You can see the 4,000 ppm NO we can get by our machine, which can be dissolved in the water for further packaging or further transport to the needful places or hospitals. We also checked pH, we also checked the lifetime, and we got that even for 1 year, you can protect 60% of NO inside this water. So you can keep a high amount of water even after 1 year. So it is very stable. For 7 days, 100% NO can be transported in the water for the treatment purposes. And many other things we measure for the physical characterization. We also check the gene. Gene are important because usually virus have nuclear gene, envelope gene, and membrane gene. So we check that how much nuclear gene are affected by this treatment. It is reduced. Even in some cases, it is more better than the chloroquine and ritonavir. And as well as E gene and M gene, same case in E gene and M gene, these are reduced. And we also check how it is affecting the lung host cell gene, antiviral response gene. So these are the lung host cell antiviral response gene which are altered by this treatment to make human cell more effective to fight against viruses. And this is about the mechanism. Basically, our plasma is targeting spike protein. So this is control, and this is, you can see, treated spike protein. So it is reducing. If it is going, the spectra is going left side, this is flow cytometer data. It means it is reducing. You can see this graph. This is control and this is spike protein after treatment. So eventually our plasma is controlling the spike protein, as well as it is inducing the host cell response genes at high level, and it is reducing the viral gene inside the cell during the assembly. So we can have nitric oxide and this plasma activated water for the treatment of coronaviruses. And this paper is already published in Bioactive Materials. Another topic is the dormant bacteria, that is VBNCs. This is very new topic in nowadays. VBNCs are viable but not culturable bacteria. So basically, whenever we treat bacteria, they become dormant, silent, and they are hibernating in soil or like environment or air or water. Every time they are available, but nobody can detect them. And the problem is that their metabolic activity is so much low that people or researchers or any lab cannot detect them by the commercial methods or the conventional methods. So we targeted in one of our study how we can control these VBNCs by using plasma. The problem with VBNC is that it is available everywhere. In environment, in soil you have, in water you have VBNCs, even in animal husbandry, or you can say livestock animal or wildlife animal also can have VBNCs. And these two things can affect our agriculture food industry also. So VBNC is also available in our food or in our drinks. And most risky population for VBNC is pregnant ladies, old people, as well as immunocompromised human being or immunocompromised people. So those are most vulnerable population that can be affected by the VBNCs, because these bacteria are dormant always, but whenever they get favorable condition inside the human intestine, they become active again and try to kill people by infecting them. So we think we thought like, can we control VBNCs by using plasma? So we checked it. You can see this plates. When we use 150 micromole of NO water, that is not very toxic. It's just biocompatible concentration. All bacteria are gone, but we don't know whether we have VBNCs here or not. So we checked these VBNCs by different experiments, new experiment we developed by using flow cytometer and other experiment. So we found in flow cytometer analysis, it is not VBNC. These are just really killed bacteria by the NO water. And we propose that our plasma strategy don't make VBNCs. Also, we checked by a specific method, that is PMX dye binding method. So that we used for specifically calculate VBNCs in our populations. And we found the bleach solution, the people use bleach solution, that is NaClO. Everywhere in all home, we use bleach for cleaning purposes. Even in hospital, it is very famous. Bleach is used for cleaning. And even in kitchen, we use bleach solution to clean our wares, vessels, as well as toilets. So this bleach is very famous, but this bleach is very dangerous because it is making VBNCs in bacteria. And these dormant bacteria available in your toilet, available in your kitchen, available in your environment, can infect your children, can infect pregnant ladies, can infect your old parents, and eventually can affect their health very worstly. So we need to think about all those things. And we compared in our study this bleach with the PG, our NO water. And we found that this bleach is making VBNC at very high consent, very high number as compared to our PG now, that is NO water treatment. We also checked some metabolism related genes that I will not explain due to time. And we checked after treatment with the NO water, whether this VBNCs bacteria can infect human intestine cell or human lung cells or other cell types. And we found that after treatment with NO water, these VBNC bacteria cannot kill our lung cells or intestinal cells. So this is very perfect strategy to control bacteria. Another one is very interesting, that is plasma nanotechnology. And it is very important for you because you all belongs to the chemistry department. So
5 minutes, sir.
Yes, yes. Just 10 minutes. Can you give 10 minutes?
Okay.
Oh,
Earlier,
okay, okay.
because we are
Yes.
already running late.
Yes, yes. So by plasma, you can make electrons. I already told you, and you can make ROS. Electrons you can use to make nanoparticle by the reduction process. In reduction process, electron played very well role. And in this kind of ROS produced by the plasma can use for functionalization of nanoparticle. So you can make many type of nanoparticle by this method. We already tested. We made a green nanoparticle, that is gold nanoparticle, without using any harsh chemical, any base, any acid, just plasma and gold solution, including dopamine. Dopamine is for coating. One pot synthesis without using much time, just 5 minutes, without using any harsh or acid and base. We made this gold nanoparticle with PDA coating, and we published in Green Chemistry, RSC Green Chemistry. In another paper, it is already going on ACS Nano. That is about the hybrid nanoparticle synthesis by the plasma by the same method, reduction by the electron and functionalization by the RONSs. And these nanoparticle can be used for various purposes such as cancer treatment or various other biomedical treatments that we are doing here. And in case of plasma agriculture, we are doing another topic that is, I will take just 5 minutes. By using plasma, we are stimulating for the seed germination and plant growth, as well as we are inactivating the microbes present in the soil or in the water to protect our agriculture part. So same machine we use to make NO water. And this is very interesting. You can also make the nitrate and nitrate, that is for nitrogen fixation in the plant for the plant growth. And we reported this work in Food Chemistry Journal recently of Elsevier. So in this case, we checked by the proteomic and metabolomics, and we found that antioxidant activity inside the plant is increased by this treatment. Also, we got very important results that this is volcano plot of how many protein are decreased and how many proteins are increased. In case of all increased protein, mostly the photosynthesis light reaction related proteins or energy synthesis related protein are increased. That is responsible for higher energy production inside the plant, and that is responsible for the growth of plant. An important part in metabolomics, we checked many kind of metabolites, more than 500 metabolites in the radish plant. This is the case of radish plant. And we found that the chemicals which are related to the quality and taste of radish, that is mooli in Hindi, are enhanced by this treatment. It means we are also increasing the commercial value that is in market of this industrial crop. And this is our future goal. We made a company last year, Placid, and this company is specifically working on the agriculture parts to have some hydroponic, aquaponic, or various other agriculture systems for the treatment and the growth. And this is the conclusion. Obviously, we can inactivate pathogen by NO water produced by the plasma. We can inactivate viruses. We can inactivate dormant bacteria, that is VBNCs. We can control the synthesis of materials. We can do the synthesis of material as well as nanomaterials. And we can do environmental friendly agriculture, such as seed germination, plant growth. And we can also increase the plant defense system that data I haven't shown here because of time. And we can modulate the energy and photosynthesis process of plants and can improve the commercial and functional, this is very important, commercial and functional values of plant products. We can increase by this treatment processes. Thank you very much. This is our center, Plasma Bioscience Research Center, and these all our team members. Thank you for listening me.
Thank you, Professor Nagendra Kaushik, sir. It's wonderful lectures, how the plasma can be useful for the virus inactivation as well as VBNC control. And also he talked about the nanoparticle synthesis and nitrogen fixation. It's one of wonderful lecture. The session open for a few questions, maybe one or two. If any audience. Any-
Feel free to ask.
Chat box, anything is there?
Chat box.
Sir, I have one question.
Yes,
You
sir.
are using NO water plasma. NO water plasma for the virus inactivation, specifically COVID virus inactivation. How this will also affect the other biological proteins and other things, because NO is known for the toxic in the biological system. So
That
how
is
it is?
actually, yeah, I will correct you here. Basically, I am biologist also with chemistry, because my PhD is from ACBR, Ambedkar Center for Biomedical Research. So basically, NO is not only the pollution. NO is biosignaling molecule in our body also. Nitric oxide is like a hormone. If you remember, whenever you have heart disease, the doctor used to give you NO-based drug to control your heart attack or to control your heart arrest. Even for the blood vessels, when your blood vessel constrict, the doctor used to give you NO-based drugs to make your blood vessels more bigger. NO is the molecule which is always available in our body as a biosignaling molecule. And it's like a hormone in our body. It controls many things. It controls it controls the hair growth. If you have some kind of less hairs, you can use NO-based drug for your hair growth. Many drugs are available specifically in market for the hair growth, as well as many internal things are there, which is controlled by the NO molecule inside our body. So NO at moderate concentration is good for our body. NO at high concentration is not good for our body. So this is the game of dose. So we need to think just about the dose. If we are using high doses, obviously NO is not good for our body. If we are using low doses, obviously NO is beneficial to our body.
Thank you. Thank you. Any other? Just last one more question. You
Yes,
talk
sir.
about the green synthesis using the plasma.
Yes.
Suppose if you don't have any, like you have used it in presence of the natural products and other things, in presence of that you are using a plasma. Suppose if you don't use a plasma, if you don't use a natural product or anything, can we get a nanoparticles? That means
No,
without capping
we are not.
without-
So that I will clear you here. All chemistry people are sitting here. You can make nanoparticle by the herbal processes also. You can use natural product to make nanoparticle. It is very famous science, very famous area nowadays. Second thing, you can use just plasma without using any herbal product. I never used herbal product. My one study is coming that in that study, we use herbal product as well as nanoparticle together to make high concentration, high quality of nanoparticle. So we can use just plasma to make nanoparticle without using any herbal product. Just we need, like in example, gold nanoparticle, just we need gold nanoparticle solution, that is AuCl4. Just put plasma there for 5 minutes and make gold nanoparticle without doing anything. No acid, no base, no natural product. But you can increase, you can enhance the concentration of nanoparticle and the quality of nanoparticle by using natural product inside it. So that is also the another indication I'm giving you all people for your research purposes.
Thank you for your nice talk. On behalf of organizing committee, we thank for your time, valuable time, and sharing your research. Thank you once again for.
Thank you, sir. Thank you very much. Thank you, madam. Thank you, sir. Thank you, all audience. Bye.
We come to the end of the session because Professor Sujit was absent. So with this, the first invited talk session has been completed. I'm handing over to Dr. Shilpi. Over to you.
As we conclude this first technical session, I would like to thank and felicitate our chairperson for the successful accomplishment of this session. To do the honor, I would like to invite Professor Harindra Chaudhary, Department of Mathematics, Deshbandhu College, to present a token of appreciation to our chairperson, Professor Shaktivel sir. नहीं Momento. नहीं, sir, this is required. You have given your precious time.